I'm Emily Miraldi, and I work at Cincinnati Children's Hospital. Hottest topic. I spoke about big data in the form of genomics data sets and how those types of measurements would influence treatment of infectious disease. I think one of the biggest questions in infectious disease is, why do some people get infected by the disease and why are other people resilient? And there are a lot of different reasons for that. So some of them are genetic. There are differences in the baseline, DNA sequence that can predispose, but then a lot of them are environmental too. And so with this genomic era, it's possible to sequence patients, for example, and try to get a sense of how genetic variation could predispose for an infectious disease. The idea there is actually by understanding how potentially patient-specific immune systems respond to infectious disease, you could change the outcome. There are a number of different technologies that are being used today that I think will impact clinical treatment. And so, for example, the advent of single-cell RNA sequencing, so we can take immune cells and actually make measurements on a very small amount of cells. There's also the opportunity, a lot of times, physicians and researchers don't have access to a lot of the tissues that are important to sites of interaction between infections, for example, the small intestine. But now it's possible with induced pluripotent stem cells, for example, to isolate, say, skin cells from a patient and rederive particular tissue types, and then in the laboratory to test how those different cell types respond to drugs or treatments or even a pathogen. So to make organoids, for example, which would be for example, of the small intestine or large intestine, and see how they respond to some kind of microbial perturbation like Shigella, for example. And so with the advent of these technologies, it might be possible to design very specific therapies for patients. When we use genomics technologies, whether they be genomics, say, looking at a particular immune cell type or tissue response to some kind of perturbation, or if we're looking at a metagenomic data type where we're trying to figure out what microbes are associated with a particular body site, we're able to come up with a lot of correlations. But we're having difficulty actually figuring out what's causal. And so I think as time moves forward and we're able to experimentally validate and test causal relationships in model organisms, we'll be able to make better and better use of these new high-dimensional data sets.